Hello, I'm Neil Spooner. I'm the chair of the Patient-Centric Sampling Interest Group, and I'd like to welcome you to today's workshop, A New Era in Clinical Diagnostics, the Power of Self-Sampling. This is part one, defining the challenges, and we're focused in North America. We've been running a similar event for Europe, the recording of which you can see through our website. Before we get started, I'd just like to say thank you to all the sponsors of the Patient-Centric Sampling Interest Group, without whose support we wouldn't be able to put on today's event. We wouldn't be able to run our monthly webinars and a bunch of the other activities we run. So thank you very much to our sponsors and our partners. So today, as I say, we're covering a new era in clinical diagnostics, the power of self-sampling. And today we're going to be very much focusing on the challenges. We start with presentations from Brad Collier, John Fabule, and Marianela Perez-Torres, who will go through presentations. I'll get them to introduce themselves in a minute. And then we'll come back for a panel discussion to include Sharon Geaghan and Bryan Andrus. So if you'd just quickly like to introduce yourselves, please. So Brad.
I'm Brad Collier. I'm a director of research and development at LabCorp. Been working with various remote sampling, capillary blood sampling technologies for almost 9 years now. Thanks.
Thank you, Brad. John?
Hi, everyone. My name is John Fabule, and I represent the patient involvement and collaboration community. I'm a physician by background.
Thank you, John. Marianela?
Hi, I am Marianela Perez-Torres. I am the director for the Division of Chemistry and Toxicology Devices at the FDA.
Thank you so much, Marianela. Sharon?
Hello, I'm Sharon Geaghan, and I am the chair of the microsampling group of the Emerging Technologies Division of IFCC. I'm also a advisory to the medical device section of the FDA, and my background is as a hematopathologist and pediatric pathologist, directing the children's hospitals at Stanford clinical labs for the first half of my career, and for the second half, the adult and children's laboratories at Stanford University as medical director.
Thank you so much, Sharon. And lastly, but not least, Bryan.
Bryan Andrus, CEO of U.S. Specialty Labs. I have been focused on patient self-sampling since 2016.
Thank you so much, Bryan. So thank you for the audience for joining us today. We're going to have 3 presentations now. If you have comments or questions at any time, please submit them through the chat function. The workshop is being recorded and will be available through the PCSIG webpage, and in fact also available through our YouTube site. But as I say, there'll be the presentations, there'll be an opportunity to submit some questions to all the participants, but also we'd like to have a panel discussion to go through what are the challenges preventing us from having more widespread adoption of patient-centric sampling for clinical diagnostics. For those of you, for next week, we're moving on to solutions. So this time, next Thursday, we'll be running a similar event, but just for 1 hour, where we'll be having a panel discussion with no presentations, focusing on the solutions and thinking through what we might want to do for future activities and who we might want to partner with to deliver those activities. So with no further ado, let's move on to the presentations. So the first presentation is Brad. Over to you, Brad.
So today I'm going to be providing a brief introduction to patient-centric sampling, PCS, and how it is helping to redefine the diagnostic experience. All right. Before I get into PCS, though, I want to dig a little bit into the existing traditional testing pathway in order to provide a foundation to better understand PCS a little bit. As you're aware, the traditional pathway starts with a visit to your doctor, upon which some testing may be decided that it is needed. Oftentimes, this testing and sample collection can be performed during the same office visit. Other times, it requires an additional appointment to be made. When we think about the sample collection, we typically think about venous blood being collected by a trained phlebotomist using traditional venipuncture techniques. And then after sample is processed a little bit, perhaps it is sent into the laboratory or shipped into the laboratory for analysis. When it is received in the laboratory, it may go through a little bit more additional processing, and then the analysis is performed. After completion of the analysis, the results are sent back to the doctor and patient, and the next step in the patient's healthcare journey are determined. All right. As you can imagine, there's a bit of clinical burden, and that produces some pain points for the patient in this existing process. The first one we tend to think about is clinical tethering, which refers not only to the patient having to go visit the doctor or go have samples collected, but it can also refer to, the scheduling of said sample collections. Then we often think about patient fear too. Those are a lot of people out there that are needle adverse. And a lot of times this comes from the invasiveness of the sample itself. Not everyone likes to have needle in their arm. Another concern we often see is volume inefficiency. We generally collect tens of milliliters of blood when collecting venous samples, and only a fraction of that sample is really needed to perform the necessary testing. And so over collection of samples can be burdensome to the patient. Additionally, it can create a lot of biohazardous waste that must be dealt with. And then the last thing we tend to think about is how resource intensive this entire process can be. All right, 2 more additional pathways we need to keep in mind when thinking about PCS. The first being point of care testing, which is characterized by moving the analysis of the samples closer to the patient and away from the laboratory. This is depicted here by having some venous blood drawn in the ED for measurement on a point of care analyzer. Remote sampling is another approach. This is characterized by collecting samples away from traditional healthcare settings. In this case, we see a mobile phlebotomist collecting venous samples in the patient's own home. After collection, these samples will be sent into the laboratory for traditional analysis. All right. There's a couple, all of these pathways, they overlap quite a bit, although they are distinct in their own right. But there's a couple other things that I want to bring up that can be a little bit confusing when we think about all of this. The first is self-sampling. Pretty self-explanatory. Patient collects the sample themselves. Oftentimes, when people think about self-sampling, they think it only applies to remote sampling, which is actually not the case. Self-sampling can be utilized for both the point of care and traditional pathways as well. Swab collections for STI testing in the clinic are a great example of this. Direct-to-consumer is another thing that's often confused with remote sampling. However, what it really is just consumer-initiated testing. In this case, the consumer being the patient. However, it doesn't have to be exclusive to remote sampling. There are cases where direct-to-consumer testing is performed via the point of care and traditional pathways as well. So where does this leave PCS? Well, in truth, PCS can fit into any of these pieces. When I was first thinking about putting together this presentation, I asked AI to put together a word cloud that describes the key characteristics of patient-centric sampling. Care is perhaps the biggest word here. Obviously, that makes a lot of sense when you're trying to keep the patient in mind when doing your sampling. Home, so that tends to lean into the remote sampling aspects of patient-centric sampling. Less, again, going back to the inefficiencies of the volumes that we collect with venous samples. Overall, if you were to dig into each of these words, what you would see is that most of them try to address some of those pain points that we brought up with the traditional testing pathway. In general, I think about 3 key components with respect to patient-centric sampling. The first, high-quality samples being collected anytime, anywhere. Also reducing the burden to the patient. And improving access and equity to the patient. And these things can empower the patient overall to provide a better experience. All right, so Purposely, before I showed the patient only having venipuncture samples, but a great example of PCS is utilizing capillary samples instead of venous samples, which I've shown here in a variety of ways in all the various pathways. A great example of this actually is the use of glucometers, fairly routine testing, but that actually represents remote point of care testing using a self-collected samples in a lot of cases. And so capillary blood is a great way to start down that PCS pathway. For this reason, there have been a lot of different capillary blood collection devices that have been developed, and there's new ones being developed all the time. But that being said, I don't want to lose sight that there are other matrices out there. Here I'm depicting our patient collecting saliva, urine, and a nasal swab here. But there are lots of other swabs that can be used also. In addition, there are a lot of other sample types that can be used. So now that we've discussed patient-centric sampling, how are we going to actually implement it? What are the key considerations? So the first one is logistics. How are we going to get kits to the patient? How is the patient going to get, samples back to the lab in some cases? How are the samples going to be labeled if we're thinking about low volume samples? There's a lot of operational questions in this piece. And then the next, compliance. What is the regulatory clearance of each of the components you're using in your kit? What is the chain of custody of that sample that's being collected? How is the data being managed for this process? Next, economics, always a big thing. We're in a new paradigm here where we're shipping kits to the patient. How is that being paid for? How is it being paid for when the samples are being shipped back? What if there's, special requirements that are needed? Lots of questions here, and there's not always clear about how it's going to be paid for. The next thing, analysis, are we using the right sample for its clinical utility? Is it accurate? Is it precise? What is the stability of the sample? Is it appropriate for our use? And then obviously moving on to the sample, do we have the right matrix? Is it appropriate for our clinical testing again? What about the volume and quality of the sample? Next, patient. Obviously, the patient falls in the middle of all of this. We want to do what's best for the patient, provide a safe and effective testing method. And a lot of times we also think about this with the self-sampling is, are our instructions good to use? There's a lot of questions here about making sure that all of this system is appropriate for the patient. And so where does that leave us today? Obviously, there's still quite a few questions out there. Perhaps my colleagues will get into that a little bit, but I think the need is quite clear. And patient-centric sampling is something that we're all going to see a lot more of in the future. And so with that, I'm going to thank.
We lost Brad. Hey, sorry about that, Brad. Cut off quickly. Thank you, Brad. We'll go on now to get a little bit of a point of view from John, giving a patient's angle on all of this. Please, if you've got comments or questions, please add them to the chat.
Hello, everyone, and thank you for joining. My name is John Fabule, and I represent the Patient Involvement and Collaboration Community United as a patient advocate. Today, I will be speaking from a patient's perspective about the need to involve the patient voice more in clinical diagnostics, and specifically the work done by the lab. The broader laboratory community being our main audience for today, while also recognizing that many others who support the diagnostic pathway are on the call. Before I go any further, I want to start with one striking observation. When I looked for data on how many different types of laboratories work with patient partners on a permanent, ongoing basis, not just for a one-off project or occasional feedback, but as a routine part of how tests and devices are developed and improved, I found surprisingly very little published evidence. And that in itself tells us something important. It tells us there is a gap. And that's across laboratories, we still do not have good evidence that this is routine practice. For example, one useful 2024 evaluation found that even in a research institute with an established involvement program, only 43 out of 83 laboratories, about 52%, had any patient or public involvement. A 2021 scoping review reached a similar conclusion, finding only 32 published reports of patient engagement in preclinical laboratory research over a 50-year period. And finally, a 2025 interview study, again, described engagement in this space as limited, with researchers pointing to the need for a broader cultural shift before it becomes normal practice. So the message I want to open with is this. We still cannot confidently say this is standard practice. Where it happens, it is still not consistent, standardized, or widely embedded. And for me, that is not criticism. It is an opportunity. Bringing the patient voice into the lab consistently and on an ongoing basis is still not common enough, but it is necessary. It is necessary because patient voice helps us understand where good science can still fail in real life. It tells us where instructions are confusing, for example. It tells us where sampling becomes difficult or may become difficult. It tells us where packaging, timing, transport, or even dexterity problems interfere with success. It shows where a technically strong test may still break down before the sample ever reaches the analyzer. And that is why one-off engagement is often not enough. When patient partnership is systematic, ongoing, and built into the development pathway, the patient voice becomes more useful, more scientific, and more actionable in my view. It becomes something you can track over time and compare against, for example, reject rates, redraw rates, usability issues, shipping failures, and borderline result interpretation. It becomes something that strengthens your quality system rather than sitting outside of it. Patients do not experience diagnostics as method validation protocol, a stability study, or an analytical performance study table. We experience diagnostic as uncertainty, waiting, confusion, delay, access or lack of access, as the case may be, and of course, sometimes as reassurance or life-saving action. And that is why I want to make this personal. And I would like to begin with a personal story, because for me, this topic is not theoretical at all. Some time ago, I developed what I assumed was an indigestion. It felt like chest discomfort, but it was not resolving over several days. It was nothing dramatic, but it did not feel right, and it was not going away. Eventually, I went to a local walk-in clinic, partly for reassurance. They did an ECG, and they checked my cardiac enzymes, specifically troponin. The result was not off the charts. It was in the high normal range for the troponin, borderline enough without the right clinical context and without confidence in the test itself. It could easily be dismissed. But because the test had been designed, optimized, and validated to be reliable, and because clinicians could trust what that borderline value might mean, I was escalated for further evaluation. Other workup showed that I had severe coronary disease and I was scheduled for open heart surgery, ACABG, cabbage. While I was waiting for surgery, I went into cardiac arrest, and it took about 35 minutes to bring me back. A less than 1% survival rate for that type of cardiac arrest, a pulseless electrical activity after 30 minutes. But I survived. And I can tell you from lived experience that the reliability and interpretation of that lab result helps set a chain of decision in motion. And that chain definitely changed my outcome. But why would the healthcare professionals trust it? And it's definitely because the lab community had done the hard work. They had optimized assay sensitivity, they had controlled imprecision, they had validated cutoff values, they had shown that a rise or fall on repeat testing actually means more than just a single static result. That is why a borderline troponin result often triggers escalation, repeat sampling, ECG review, clinical correlation, and sometimes urgent cardiovascular evaluation, rather than simple reassurance and discharge. So when I look at lab teams, I do not see people who are far away from patients at all. I see people who are quietly and often invisibly help decide whether patients like me get a second chance. And that is why the work the lab does matters and why it matters so much to me. The way I picture and explain the connection between patient sampling experience, patient voice, and work done in the lab is this. I'm reminded of patients boarding a plane when they collect a sample at home. They are busy, they may be anxious, they may already not be feeling well. The pre-analytic variation is the weather. That variation might be delays, heat exposure, underfilling of samples, mix-ups, or even hemolysis. The method and quality control are the control tower. Those responsible decide what is safe to land. In other words, what is safe to report and what needs to be rejected and redrawn. The clinician is the pilot making the final decision. And of course, the patient voice is the weather report. It tells us what conditions are actually happening in real life. And that is what makes it so valuable. So why does the patient voice matter? Here is a key point. Even a perfect essay can fail in real life because of what happens before the sample ever reaches the instrument. Patient voice matters because it helps us understand and reduce real-world failure points. This might be underfilled samples, mislabeled specimens, delayed shipping, temperature, exposure, contamination, poor mixing, incorrect timing, confusing instructions. And these are just some examples. Or very human barriers such as pain, embarrassment, low vision, language barriers, or even limited dexterity. When those things happen, they do not stay invisible at all. They show up in the lab as more rejected samples, more redraws, more inconclusive results, and more noise in the data generally. So, for me, patient insight is not about relaxing standards at all. It is about making it more likely that real-world samples actually meet the standards you already require. So the patient voice does not weaken quality. For me, it strengthens quality. And I know when I say the lab, I know that means many different things. Because some labs are trying to discover new science, some are building or validating new tests, some are supporting clinical trials, some are running hospital testing every day, and some are helping patients use testing closer to home. So I'm not saying every one of those labs needs the exact same patient role. What I'm saying is that whenever the patient experience affects whether a test is used properly, whether it is trusted, understood, or even completed at all, the patient voice matters. Across the field, we still do not have strong evidence that ongoing, built-in, permanent patient partnership is standard practice. And that is why this matters. Because if patient voice is only appearing at a few touch points, lab may still miss the real-world problems that decide whether a good test or device succeeds or fails. Common question that often come up. First one isn't patient voice subjective? How can we use it scientifically? And my answer is this. Treat it like any other input. Collect it systematically, look for patterns, and convert those patterns into testable conditions and measurable metrics. For example, such as reject rates, hemolysis rates, insufficient sample, or delayed shipping. For example, if 7 out of 10 patients say, I can't tell when I've collected enough, to me, that is not just an anecdote. That is a design signal. And in the lab, that signal may show up as insufficient volume rejections. So what do you do with that? The design could be addressed. The level could be made more obvious. That insight could be converted into validation work. Low volume samples could be tested, for example. Reject thresholds could be defined. Instructions could be updated. And then the effect on insufficient volume rejection rates could be measured. And that, to me, is how subjective experience from patients can become scientific improvement. Another question we get is, how do we stop this becoming a never-ending wish list? And I think that's a fair question. And my response to that is to set boundaries. You can say, for example, we are focusing on changes that improve reliability, reduce burden, and expand access without compromising analytical vigor. Our patients usually respect those kind of boundaries when they are explained clearly. So, for example, a patient may say, I want results immediately, or I wish these involve fewer needles. Now, the exact request may not always be possible. Some limits are set by the biology of the test and by what is needed to produce a reliable result. But the value of that comment is that it shows you where the burden sits for the patient's point of view. So it may not mean you can remove every step, but it may mean you can make the process less confusing, less stressful, and easier to complete correctly through perhaps clearer instructions, better timing guidance, simpler kit design, or more intuitive packaging. And these are just examples. And in summary, you could just say, we listen broadly, but we act narrowly on the highest risk and highest frequency failure points. The third common question we get is, for those who are focused on assay validation or method development, how can patient insight realistically change validation plans? And to that, I say, patient voice tells us what real life looks like, so we can validate the method under the condition where small signals must still be trustworthy. So going back to my own story, cardiac troponin made a difference to my outcome, even when the result was in the high normal range. And that tells us that borderline results matter, especially when the assay has been optimized and validated at that level. So what can someone in assay validation or method development do? Add pre-analytic stress tests based on what patients say actually happens. So for example, room temperature versus a warm car, mild hemolysis or lipemia threshold, repeatability at low concentration near clinical decision points. These are just few examples. And we do get other questions on how many scenarios to test for and patient influence on interference panel, but I won't go into details about those due to time. Let me move quickly to something more practical, which is what does patient voice actually look like in the lab on a day-to-day basis. Patient engagement, for me, does not have to be large or complicated. A few focused actions can already make a real difference. And that may be where you start your focus. So you could, for example, organize a small patient partner group, do a sample journey review and ask patients to walk you through collection on their worst day, not their best day. Observe a usability run through before you lock the method. Add patient's reality into validation by including delays, temperature swings, underfills, or shaky hands, and then quantify the impact. Co-create instructions in plain language while preserving the scientific requirement. And one that often gets left out, which I will definitely encourage, is to include collaborations with patients in publications. And if you think time is short, I think the two places I would recommend starting are sample journey review and patient reality and validation. Those alone can go a very long way. Another common question is this: we do not have access to patients. How do we start? My answer to that is, start with what you already have. Look at complaint logs, for example, if you have them, or redraw reasons, call center questions, and even clinician feedback. That already tells you quite a lot. And then, of course, you can then move on to partnering with an existing patient group or network where governance is already in place. For example, patient communities like PiCC United or Patients Like Me. I think the overarching principle, which is basic, is this: engage ethically, transparently, and with a clear scope in mind. Patient voice works best when it is structured, respectful, and used to drive measurable improvement. So I'll close with this. Patients do not experience diagnostics as a technical process. We experience it as worry, hope, delay, clarity or lack of clarity, as the case may be, and sometimes as reassurance or even life-saving action. To me, labs teams are central to that story. When you optimize an assay, define stability windows, reduce pre-analytic errors, clarify instructions, improve acceptance criteria, or refine threshold, you are not just supporting patient care, you are enabling it. So my invitation today is simple. Build patient reality into your quality system in a systematic way, not as a campaign, but as a routine, measurable partnership. Thank you very much for your attention.
Thank you, John. Thank you for that very insightful presentation, putting the patient at the center of all our thoughts here. I think that's really important for our ongoing conversation later on. But without any further ado, I'd like to move on to Marianela's presentation, and then we'll move on to the Q&As. Please don't forget to submit any questions through the chat function.
Hi everyone, I'm Marianela Perez-Torres, Director for the Division of Chemistry and Toxicology Devices at the FDA. Today, I will be presenting on the regulatory landscape for self-sampling collection devices and share some of the questions that FDA considers during the pre-market review of these devices. The presentation is intended for informational purposes only and does not constitute legal or regulatory advice. Also, please note that examples referenced in this presentation are for educational purposes and do not represent in any way FDA endorsement to any specific device. In this slide, I will be describing at very basic level the regulatory framework for medical devices in the US. The FDA classifies medical devices, including sample collection devices, into one of three classes, based on the degree of risk they present and the level of controls that are necessary to provide a reasonable assurance of safety and effectiveness of the device. Device classification depends on the intended use and upon the indications for use. The class to which a device is assigned determines, among other things, the type of pre-market submission required for FDA authorization to market. As device risk increases, the classification and the regulatory controls also increase. Let's start with Class 1 devices. These are considered low risk and are subject to general controls, which are regulatory controls to which all devices are subject to unless exempt by regulation, to ensure their safety and effectiveness. General controls include provisions of the Act pertaining to adulteration, misbranding, device registration and listing, labeling, and good manufacturing practices, as set forth in the Quality Management System Regulation, which I intentionally highlighted in a separate box to emphasize its importance. Most Class 1 devices are exempt of the pre-market notification procedures, unless it's considered a Class 1 reserved device or exceeds the limitation of the exemptions, like new indications for use or technology that is different from what is described in the regulation. To be clear, manufacturers of Class 1 devices are required to validate them based on their intended use. While they don't need to submit any file to FDA, they must keep records internally, as this document can be examined by the agency during site inspections. FDA classifies into Class 2 devices with moderate risk, for which general controls alone are insufficient to provide reasonable assurance of safety and effectiveness, but there is sufficient information to establish special controls to provide such assurance. Special controls are usually device-specific and may include performance standards, post-market surveillance, specific labeling requirements, or pre-market data requirements. Most Class 2 devices require a pre-market notification that is known as 510K, where companies demonstrate that their devices are as safe and as effective, that is, substantially equivalent to a predicate. The FDA classifies into Class 3 devices intended to be used in supporting or sustaining human life, or that present a potential for unreasonable risk of illness or injury. Most Class 3 devices require pre-market approval, or PMA. This is the most stringent regulatory pathway and includes, for example, review of manufacturing information. Before FDA approves a PMA, the sponsor must provide valid scientific evidence demonstrating reasonable assurance of safety and effectiveness for the device's intended use. Devices that didn't exist or were not known before the date of medical device amendments of 1976 and have not been classified by FDA are automatically classified into Class 3 regardless of the level of risk for that product. However, in 1997, the de novo path was established and enacted to allow novel devices to be classified into Class 1 or Class 2 if general controls or general and special controls provide a reasonable assurance of safety and effectiveness when there is no legally marketed predicate device. The de novo review is a risk-based classification process. During this process, FDA assesses the data and determines if the probable benefits outweigh the probable risks and the level of controls needed. If the de novo is granted, it establishes a new device type along with a new classification. Also, if the de novo is granted, the device is eligible to serve as predicate for new medical devices where appropriate, following the 510K process. Here are some examples of different types of sample collection devices and their current classification. Many basic receptacles, like simple urine cups, are Class 1 and often exempt from submitting a 510K. Another example are the straw-like capillary tubes that are part of the manual hematology devices under A64 Subpart G of the Code of Federal Regulations. As mentioned before, exempt devices are subject to limitations of exemption, such as a new intended use or new technology. For example, Class 1 sample collection devices that are legally marketed only for professional use would exceed the limitations of exemption when intended for self-collection by a lay user. In those cases where the limitations of exemption are exceeded, a 510K pre-market submission is expected. Devices like blood collection tubes and dry blood spot cards used for downstream testing of analytes, or non-invasive collection of oral fluid samples for clinical DNA testing, and microbial nucleic acid storage and stabilization devices are Class 2. There are also a few examples of sample collection devices that are regulated as Class 3, along with the high-risk tests they are intended for. One example is the swab used for taking a vaginal secretion sample used to detect a biomarker that aids in assessing the risk of a spontaneous preterm delivery. There is also a blood collection tube intended to stabilize circulating cell-free DNA isolated from cancer patients. This sample is currently regulated as a component of the corresponding liquid biopsy test. FDA has authorized several collection devices for self-collection at home, and here are some examples. A device intended for collection of saliva samples for testing of human DNA was cleared under the 510K path. A device intended for self-collection of vaginal specimens at home or a private setting for use in HPV testing was authorized through the de novo classification process. And a dry blood spot card cleared to specifically collect samples at patients' home and be tested with a specific A1C test was cleared through the 510K pathway. In this case, the DBS is part of this specific test and is not clear for home use separately from these tests. We want to see additional successes in this space, but there are fundamental gaps in knowledge that will be helpful to address in order to see major progress in this area. From this point forward, I will focus on questions that we have to ensure that self-collection of capillary whole blood samples at home using new technologies are safe and effective for their intended uses. The first challenge in this space is the sample type. In general, capillary whole blood is a complex mixture of arterial, capillary, and venous blood, along with undetermined proportions of interstitial and intercellular fluid. Because of this mixed composition, the concentration of certain biomarkers may differ from a standard venous draw. For example, for capillary samples collected from the finger, it is known that glucose levels are higher than in venous samples, and also that capillary samples that have lower level of total protein, calcium, and hematocrit when compared to venous. However, it is not well established that capillary samples collected from the arm yield results for all analytes that are equivalent to the venous samples that have been typically used to validate the FDA-authorized test. To move the field forward, the entire industry will benefit from generating scientific evidence to better understand equivalency between matrices. Now, let's take a look at the devices that are part of this growing ecosystem. The first generation of open arm devices integrated the tube for collecting, storing, and transporting the whole blood. However, the newer versions have split the lancing device from the receptacle. In the current landscape, open arm lancets that are FDA cleared have no compatible tubes intended for open arm sample collection, and have not been cleared for use as a sample collection device. Then, the capillary collection tubes are currently not cleared by FDA for use with the open arm lancing devices or open arm capillary collection, and are intended only for professional use, not for self-collection by lay users. Dry blood spots and devices that use other absorbing materials pose a unique challenge because these require additional steps of drying, punching, and extracting the sample using buffers that deviate from the plasma or serum that would be generated from a liquid blood sample. Most tests that are cleared or approved for testing in large clinical chemistry analyzers have been validated using samples collected from a vein, and also have specific recommendations on sample handling and storage in their labeling. Sample collection devices need to align their instructions and handling conditions with the current labeled conditions of the test. Further, because the test manufacturers did not design their device for capillary blood nor for blood reconstituted from filtered paper, the design control processes and their quality systems do not address nor control for this mode of testing. Ensuring who assumes the regulatory responsibilities, including medical device reporting, recalls, design controls, for each piece of this ecosystem is vital to ensure safety and effective use of all these devices. Considering all the pieces in this puzzle is very important because it is well known that the performance of blood collection devices can impact the test results. For example, the storage conditions, including shipping conditions, of collection devices prior to and following blood collection, as well as the materials used in the manufacture of the device and the time of contact of the specimen with those materials, can adversely impact the sample quality and increase the analytical error of downstream testing. Once the fundamental question about matrix equivalency is addressed, there are additional considerations regarding the sample and its successful collection at home. Here, the operator and the environment matters. While we understand the desire for providing patients for options to collect samples at home, we also appreciate that validating the use of these devices at home is quite challenging. Even for professionals, collecting a sample from the finger can be difficult. This is because capillary blood clots much faster than venous blood. If the blood isn't quickly mixed with an anticoagulant in the microcollection tube, microclots will form and the sample will not be suitable for testing. The red blood cells in a capillary sample are highly susceptible to hemolysis. This is often caused by the mechanical stress of squeezing the tissue to force the blood out, or by the shear force of the blood being pulled through a tiny microneedle or a vacuum channel. It will be very important to demonstrate that lay users can collect a sample that is of sufficient volume and quality. Studies should include a diverse representation of the intended lay users. This means accounting for different levels of education, health literacy, ages, visual acuity, and physical dexterity. Users should be able to do the sample collection process without any coaching or prompting to represent real use of the device. As for the environment, self-collection will likely happen in a home bathroom, kitchen, or a living room. Validation should consider correctly representing or simulating environmental factors like poor lighting, lack of a sterile surface, and distractions, which are very different from a clinical setting. The manufacturer should also consider mitigations needed in this environment. For example, the users may fail to properly sanitize the puncture site with alcohol, risking localized infection. Ideally, the device will incorporate a fail-safe mechanism to prevent accidental needle sticks to the user or family members. Many analytes are uniquely affected by continued contact with the cellular component and the temperature conditions after sample collection. Based on this, a 2-hour maximum delay before separation is recommended for most specimens, and such separation is typically conducted before specimen transport. It would be very important to demonstrate that analytes in samples collected at home are stable in whole blood during the worst-case scenario transportation period and conditions. As mentioned before, if intending to use a previously cleared or approved assay, the shipping conditions should align with the recommendation in the test labeling. Whole blood is highly sensitive to temperature fluctuations. Therefore, shipping stability studies should evaluate the device function and sample quality after the device is shipped as intended and exposed to all the extreme environmental conditions that are possible during this process. The study should simulate conditions that are commonly encountered across the United States, including ambient temperature and humidity, extreme cold, extreme heat, extreme humidity, light exposure if intended for measuring photosensitive analytes, and vibration that may lead to device failure, like sample leaks. Other challenge during validation of capillary samples is that the volume is too small for testing all the analytes that the device is intended for. This may require collecting multiple samples while accounting for potential biological differences in analyte hemodynamics, or enrolling additional patients to collect sufficient data. We acknowledge that the difficulties to generate the data to support safe and effective use of these devices are not trivial. But if the ecosystem have the right stakeholders finding solutions, it is possible to see progress in the near future. Thank you for the invitation, and I look forward to our discussions today.
Thank you, Marianela, and thank you to all the presenters. If we can ask all the presenters and the panelists to come on stage, please. Welcome back, Brad, John, Marianela, and welcome again, Bryan and Sharon. We've got an opportunity here to talk about the challenges, but first of all, I'd just like to go through a couple of questions that have come through. I think, Marianela, you've got a few questions here. Maybe if we can answer them quickly and then move on to what the challenges are. But for you, a question from Valeria to you, I think, Marianela. How do you differentiate between low, moderate, and high risk? You're muted. Yeah.
I'm trying to unmute.
You're okay. I can hear you.
You can hear me?
Yes.
Okay, great. So basically, most of the devices, when the amendment to the Food, Drug, and Cosmetic Act was made in 1976, soon after the classifications were made based on the devices that were available back then in the time. So it has evolved our thinking over time. So for the new devices that have not been classified before, we look at the risk, and usually if they support human life, it is typically PMA, as I already say. The boundaries between class 1 and class 2 can be more blurry, if you will. So here is not a one-size-fits-all. Details matters regarding what the device is and the clinical indications and the risk with those clinical indications.
Okay, thank you. I realize there are quite a lot of questions for Marianela, but what I want to focus on today is not too much on this being a Q&A session in a traditional sense, but the idea of what we're moving towards here is we've seen presentations about what these technologies are, what the potential are from Brad, presentation from John about how we should include the voice of the patient and when we're moving forwards, and a great presentation from Marianela about the challenges we have ahead and the things we need to consider in order for the devices and the data to be accepted. So what I'd like to get into before, if we get time to come back to some of the questions that have been posed, we'll do that later. But what I'd like to get into is, if we're seeing there are benefits to these technologies being used for clinical diagnostics, what are why aren't we using them? What are the main challenges that are stopping us from moving forwards? I don't know whether anyone has an opinion they'd like to start the conversation with here, but what are the main reasons that are stopping us doing this?
Yeah, yeah. I mean, I can start from a patient perspective. I think having the patient confidence in using the test is very critical. Because if a patient does not have any confidence that the test is going to do what it's supposed to do, or they will need to repeat the test again after going through the inconvenience of pricking themselves or having somebody take the blood, then the patients are not going to use it, and that's going to translate into adoption. And because if patients are not using the test, then payers may not be interested in paying for it, and so on and so forth. So for me, having patient confidence in that test, not just the kit step, but the result of the test, is quite important.
Thank you, John. Would anyone like to build on that at all? I think it's great that we started off with the patient, so that's important. Yeah, sorry, Brian.
Oh, that's quite all right. I agree with John. I think that patient confidence, provider confidence is critical. Candidly, I was disappointed that the LDT governance had been dropped because I think it's very, very important for anybody that is part of this ecosystem to be able to stand up and know that there's equivalence with the tests and that people are following the same sort of guidelines with respect to validation, and that validation also includes the components that we're touching on. I'll spend as we millions of samples coming in from patients since open the lab. And
Brian, sorry to interrupt. Can you move a little closer to your microphone? You're breaking up a lot.
Oh, sorry about that. I hope I'm not yelling at people now. We've processed millions of samples that have come in from patients, directly from patients, and we have done everything we can from a validation perspective. Thermal cycling, vibrations, everything is part of our validation process. And I think it's important that the laboratories and the ecosystem that is supporting these types of samples in the field are following the same sort of good practices, because otherwise people are not going to, we don't need to talk too much about Theranos, but I'm afraid there's going to be another Theranos moment with some of the ways that people are playing it fast and loose with some of the microtainers. And there's a good way to bring those in, and there's a way that is not so good. And unfortunately, there are people doing that. We just need to really self-police.
So you think that the Theranos kind of problem is still a big barrier in North America?
Well, we're asked about it quite often. It's mainly from lay people that are just starting to take a look at the adoption of this, but it's out there, and I think it builds on John's, or maybe John's builds on it, the concern about equivalence and confidence in it. I mean, that really shook us to the core. It was a big hit for us. It was not something that we were interested in having to deal with for several years.
And so do you think that's building into patient reticence and regulatory reticence as well?
Well, I'll let Marianela speak for the regulatory side of it. But and John can speak for the patient more accurately than I can, but I think it all combines. I think people want to know that they are going to be getting an accurate lab test, and they need to be confident in that. And we encourage people to do blind testing, for the samples that they take as they're starting to come on board with us.
Yeah, no, I will agree because everything is all interconnected. If patients have confidence in a test, then they're more likely to use it. Then the payer is more likely to pay for it. Then the regulators are more likely. So it's all interconnected in my view, and it's the whole of pathway works hand in hand.
Okay. Sharon, Brad, have you got anything you'd like to comment on?
I could. I think that what we're asking laboratories to do to a large extent is treat, very small sample size, really like pediatrics. And, the drivers for cheaper operations is very strong. And what you do is with a very low volume sample, you have the problems of not being able to use automation. You have problems with, aliquoting from the sample, a lot of manual manipulation, sort of like undoing a lot of the efficiencies that, the robotics and lab automation have brought. And that requires more time and a staffing model that supports it. And it's expensive. And, that's not only true for the US, where there's, private insurers. The government is still the biggest payer, but also in Europe. I mean, it does boil down to a different staffing model and more labor cost. So in terms of an obstacle, I think the economics of it have to somehow be factoring in, like, how does this work in the whole cycle of the journey that John was outlining, and what are the ways that this actually saves, because you have to build an economic case, I think, that's compelling, ideally.
Yeah, no, I completely agree. And do you think that actually sort of has to do with the fact that we are not usually very good at measuring the patient burden as we are with measuring the lab burden, so the workforce in the lab and the operational feat for certain things in the lab. But for patient burden, like worry, like confidence, like, we are not very good at mentioning this. And if you can measure them, then there's no evidence behind that. Then if you can do that, then payers are, saying, show me the value for me to pay for it. So do you think those two are connected?
Yeah, I mean, I know that there are different models with remote sampling, for example, in Africa, where a person might be taking HIV meds, they need TDM. You can't really responsibly prescribe these medications or seizure medication without monitoring them with TDM. If they have to take, buses to a regional hospital and take off 2 days of work and back and so forth just to get that one test, that's a really compelling economic driver for, moving it closer to the patient, whether it's remote sampling, point of care, and so forth. But, I'm imagining that the industry players have models that they put forth in terms of how they see this being advantageous. But it's hard because the actual cost center, of course, is the hospital, and that's what they're interested in. So yeah, it's very tough, but definitely from the patient point of view, there's all kinds of savings.
I'll layer on with the cost that you're speaking to, Sharon. You mentioned the government. We think about reimbursement codes a lot. There aren't any reimbursement codes for kits that are shipped to patients, for instance, or shipping back. What about additional tools that may be required if there's like special shippers that are required to make sure we don't, harm the sample in any way? These are all other questions that need to be considered. But I also want to thank Bryan for pointing out all of the various analytical studies, as I tend to come from that side of the things.
Thank you, Brad. So we're going down some very similar directions to the European discussion, actually, which is not surprising, I guess. I thought there might be some differences with different regulatory oversight, and maybe some more proximity of the Theranos thing is here, but it's very much coming down to patient confidence, which leads through to regulatory confidence, I guess. Lack of automation and cost benefit models is the three things I'm really hearing here. Are there any other big barriers? Am I missing anything there in distilling it down to those three main ones that we've heard so far?
May I, Neil?
Yeah, absolutely, Marianela.
Yeah, no, I don't want to be perceived, FDA, be perceived as a barrier. We are gatekeepers of the medical devices that come to the US. So we base our decisions on data. Patient preferences are very important for us, and our center have issued guidances regarding how to incorporate patient preferences in the design and the considerations that we take into account when we review devices. So that's important. The regulatory skepticism maybe doesn't come from one company or necessarily for public perception. We take it into account, but it's mainly driven by the data that we see that is not always available to the public. So as long as the data that we receive for these devices is solid, is credible, is generated in a controlled way that demonstrates that it works as intended, that's all we need. And I see it simply, but it's a difficult task for manufacturers to do.
So
right.
Are you suggesting perhaps more transparency in the community with respect to things they're observing?
That will be helpful for us because it's information. We look at the data that manufacturers submit in the pre-market package, but at the same time, it's our same team monitoring the signals for the devices that are already in the market. So the post-market signals, we manage the recalls, we manage malfunction reports, and that information is used in our total cycle review, where signals that come from the post-market side is incorporated in the pre-market side and helps making better the type of devices that come out. So yes, transparency is important, and having the information. We're here. I guess my message is the regulators are here to help. We part of our job is to promote public health, and we promote it by giving access to the U.S. population of the technologies that they need to improve their health. That is important. We're not here just to go after companies. We're here to make sure that we provide for the public what they need. And that's the message from us. We're here to help. We want to find solutions. And I know that solutions are for next week.
No, absolutely, Mariana. But I think that what you're just saying, Mariana, leads into a comment from Gloria, part of our audience here. I'll just read out what she says. One thing that I'm confused about is that there are so many home collection test kits, like you can walk into a drugstore like Walgreens and get any number of kits that collect your blood and send it to the labs for analysis. Can you help clarify how this is allowed by FDA? There have only been warning letters about HIV home collection from DBS. So yeah, that confusion over, you were showing the lack of approved devices and tests, yet people can walk into Walgreens and get these tests.
Right. And without getting into specifics of the warning letters that were referenced or the HIV test in particular, because those devices are regulated by our Center for Biologics. It's not the Center for Devices, but we do have a relationship with our colleagues. Many of these things, that's part of our post-market activities, where maybe some manufacturers may perceive that they can use those kits and bring them to the market, and because they don't import from other places, they just go ahead and do it without having our blessing. And once we become aware of those issues, we take actions to protect the public from things that have not been validated. But doesn't mean that everything is everything that is on the shelves have FDA authorization. And if I may, I saw another comment that was referring to our class 1 devices being registered and listed in our database, and there's confusion regarding why those class 1 devices may or may not be used for downstream testing. So clarification number 1, that registration and listing database, if is managed by the companies, so they self-register their devices based on their intended use and the initial classification. It's not a database that we curate constantly. Once we become aware that something is listed erroneously, then we intervene and we contact those companies. But as you can imagine, there's so much work to do that it's impossible to cover all the bases. So we also rely on the honor system that the companies are following the law. And when they have confusion in interpreting the law, of course, we're here to help them understand what it means.
Thank you, Marianela.
Marianela, can you speak to the concept of laboratory developed tests?
I cannot today. That is a topic that, based on the litigation results, we are unable to comment just yet.
Okay.
But going back to this discussion, thank you, Marianela, thank you, Bryan, with the kits being available in drugstores, I guess that leads back, Bryan, to what you were talking about to a certain extent, the danger of, it sounds like, unregulated tests being available, potentially not giving high quality data. Therefore, that gets out there, and all the good quality devices, good quality assays that have been properly validated and qualified are then, we end up throwing, as we have the phrase, throwing the baby out with the bathwater. We end up throwing the good things out because these bad things have tarred that. I don't know whether you want to comment a little bit more around that, Bryan, because I know it's something you believe in strongly.
Well, I definitely believe strongly in the need for, as I mentioned when I spoke previously, I was disappointed that the FDA had declined to jump in and, follow through with their stated intention to become involved in laboratory tests, because I think there needs to be some additional structure on that. And I know I'm going against all kinds of associations and probably most of the other laboratories by saying that. But most of the kits that are out there, it is being done through laboratory developed tests, and as Marianela says, it is on the honor system at this stage, with the FDA not coming in on it, that these tests and assays are being validated properly. And, Brad and I've had some conversations about some of the ways people are handling the return of samples from the field, and how that does impact the clinical viability of the samples. And we really do need, as a ecosystem, to certainly self-police. And I would, again, welcome additional help. I know it's good. It will be good for the adoption of these devices if there could be some level of oversight coming in on it. So, and then Marianela, that's why I asked the question about the LDT. Not that I'm challenging you. I want to embrace the fact that we could use that.
Yeah.
As Brad's head bobbing up and down, I think he and I are very much on the same place there.
I guess I cannot comment on the LDT ruling and how it refers to the actual test. I think it was clear from that blood collection devices themselves are not LDT. I think that's part of what appears to be, but that's all I can say today.
Yeah, and LDTs are, they are a valid way of going about performing a test. And they are, as long as the laboratory is adhering to standards and, CLIA, CAP, CLSI, whatever the guidelines that are being used, or internal standards, then they can show functional equivalence, clinical equivalence between, matrices and predicate, similar to what you would do, Marianela, with when you're looking at other devices. That's on the labs. The labs are obliged to do that. And,
right, but in this space, if there are self-collection devices that have been clear for broad use rather than the tiny pieces that we have been able to authorize today, that will give more tools to the lab to implement other things, right?
Absolutely.
So that ecosystem. We have a manufacturer of a test that didn't think of these other alternate devices for them. And then we have pieces that are on their own place, but they have to come together in order to generate the data.
As you were both talking, I was thinking about this is a system that we're validating. It's not just an assay, for instance.
Right.
It's inherently interconnected, I'm afraid.
Yes, there are pieces that have to be considered, and there are various, regulatory approvals. How are you going to validate that, whether it's internally or via regulatory pathway? It's more complex than just developing an LDT because of the nuances of taking it outside of the laboratory and how the samples are collected.
Mm-hmm.
Yeah, and I think some of the manufacturers are following the regulatory pathway to add different sample types as part of their IFUs, and are starting to do, we're working with some that are starting to do the studies that will be submitted back into the FDA to expand the different sample types, as long as there's equivalence on that for the reagents that may be used as in an FDA cleared environment. Doesn't mean the test is necessarily, but that's something that we're looking forward to seeing more of.
Us too.
Yeah, I'll bet.
I just wearing my pediatric and neonatal hat. There you're talking about a lot of off-label use, and it's not unique to lab. It's true of pharma too. And, since we're talking about obstacles, it's something that I think that in the pediatrics arena and neonatal arena even more, the collection devices we're talking about, they have not been validated on children. Even the ones that, have made that hurdle with the FDA. So that's true. That's tough too, because some of them have great advantages with the cognitively challenged and those that have disability around understanding what's happening to them with phlebotomy. We've had children that are very disabled cognitively, and it's really hard to get blood draws from them. And it may take a lot of time and a lot of work, and they still can't get blood draws, and they need them. And they're taking a lot of medicines, perhaps for seizure disorder or whatever. And some of these new devices have advantages. Because if you have a needle phobic child or what have you, you'd like to be able to use them. But again, it's off-label.
Mm-hmm.
I don't know those of you that have seen it or not. We've been putting it through on the PCSIG website and LinkedIn, whether you've seen Abby's story, which is story from a child with Down syndrome in the UK who found it very stressful giving blood samples and has been using an upper arm collection device. And there's a film, we have a film with her mother and the child. And in this region of the northern UK, they did a test, and suddenly people that have very rarely had a blood test or never had a blood test are now being tested regularly. And they like blood testing. Abby describes this as fun, whereas standard phlebotomy, she was very averse to. And this was put through Facebook and other places, and there's been just a huge response. And we're going to be, PCSIG are going to be helping with a follow-up event to just bring together patient groups, payers, others, to just see how this excitement for these underserved communities can be helped. And I'm just wondering, we're busy talking about regulations and validations and rules, but does any of this take into account the communities that are currently underserved, the people in remote areas? As Sharon was talking about, children that have never been sampled, they don't care. They just, any test is better than no test, you could argue. Is there anything to be thought about there?
Well, thanks for bringing that up. Our colleague Karen Perkins has really been a champion in the UK,
Yes.
for bringing that all to light. But I guess we're getting to the question of how, also analytically, how good is good enough. And I think that we really need to build the bridges with patient-facing clinicians, because sometimes, again, it doesn't have to be quite as good. I totally respect the FDA rules about predicate and about, I think it's very sound. But on the other hand, like, at what level could a test be not quite as good, but perhaps in situations like remote sampling situations and so forth, it may be good enough. And
or
I think
in the
that's
absence of other testing.
Yeah, exactly.
It's in the context of the intended use and looking then,
Right.
besides substantial equivalence, then benefit risk. So that's where there's a benefit of self-sampling, there's
Yes.
risk, how we balance them out to allow
Yes.
a device that is fit for purpose. And it's something that we can work with industry on. No device is perfect.
Right.
We will clear or approve nothing if we were waiting
Yes.
for perfection, but it's how much we can tolerate and how we ensure that it's used in a way that the risks are mitigated and patients are safe to do that. So it's
Right.
finding that right balance. And we are happy to work with manufacturers on doing that.
We're glad you're there.
So maybe just one last area. I mean, are there any other barriers to adoption? We talked a little bit about laboratories and lab automation. We've talked a little bit maybe about cost benefit models, and as well as the regulatory space. Is there anything else we're missing here that we need to maybe pick up on for talking about solutions in a week's time? Or are they kind of the three main places? Difficulties in the lab, patient acceptance, cost benefit models, regulation, etc.
Logistics is always a bit of a problem, but that kind of incorporates the automation part. But I would expand that to logistics of, getting kits to patients, making sure they don't arrive damaged, getting it back from them in a high quality manner. That's good for testing. That's a piece that's probably missing. Sorry about the puppy.
Data management.
Yeah.
Yes. Yeah. I mean, getting the data to the right people, knowing who the data belongs to. Yeah, that's a really good point, Sharon, and a big gap. We're kind of running out of time here. We're at the end of the hour. I think that's been a really good discussion. It's exactly what we were looking to precipitate. I thank you all for an open, honest discussion. We've covered a lot. If we can just get, if I can get the slides back up. So as I said before, this workshop is being recorded and will be available on our website pretty soon. Just to go through the next steps, we're going to be compiling the output from today's workshop. So we had an AI tool recording this. I've been taking notes, and obviously there's all the comments from all of you out there. Apologize if we didn't get to your comments. We will be taking note of them and incorporating them into our discussions. And basically we'll be compiling everything together, be sharing that with the panel, and we'll be prioritizing for part 2 of this event, which happens on the 7th of May. At the same time, it only goes on for an hour. And on that occasion, we're going to be prioritizing some of these challenges, and we're going to be talking about what the solutions might be. So how can we actually move forward with these tricky situations that are stopping us having this kind of home testing, these home collected samples with laboratory tests being more widely available in North America. So we're going to be talking about that in a week. Those that have already attended today, you're already registered for next Thursday. So you will receive a calendar invite for that. So, but for those that are watching this on catch up, the registration page will be on our website if you weren't actually here live at the event, but you'd like to attend the follow-up event next Thursday. So that registration page will be on our website, and we'll be putting out the content of this event through our LinkedIn group. For those of you that don't know the Patient-Centric Sampling Interest Group, just these QR codes, the one on the left will take you to our website where there's lots of interesting materials, lots of webinars. And we're always interested in people joining our working groups. On the right-hand side is a QR code to take you to our LinkedIn group, where please join the ongoing discussions and sharing of information there. As I say, part 2, we'll be discussing solutions, and then we'll be bringing the panel from the European event and the North American event together to discuss what do we want to prioritize as next steps. So how are we going to share with you all the output? Is that going to be a publication? Is that going to be a webinar? Is that going to be content on our website and LinkedIn? And will we have any working groups? I suspect we will be having working groups. If we are, we need to think through who we want to partner with, and then we'll be coming back to you, PCSIG community, to take part in those discussions and contribute to the solutions and to help us all move forwards. So I'd like to just thank our partners and sponsors again. I'd like to thank all of our speakers and panelists today. I think this has been a wonderful event. I'd also like to thank all of you out there for attending. It's really important that we have an audience. I think this is an exciting and important area. And so please join us and help. Thank you.